Hey everyone, my name is Dr. Dominique Lessard, and I'm the Chief Science Officer here at Kifone.org. And one aspect of my job as CSO is to be a leader in our therapeutic development strategy as we work towards finding a treatment and a cure for CANT. So today, I'm going to tell you a bit about the therapeutic development process, inform you of where we are at today, and discuss plans for the future. Let's start out with a basic question of what is therapeutic development? Now, you can type this question into a search bar and you'll likely find a definition that says something like this that therapeutic development is a collection of preclinical strategies to support the development of products intended for use in the cure, mitigation, diagnosis, or treatment of disease. But what does this definition really mean? And importantly, what does it mean in the context of what we do at kifone.org? We can reframe what we're doing as a question, and we can ask, how can we gather information in a laboratory setting, in a scientific space, and use that information to develop treatments for CAN? And in answering this question, we are working towards our common goal of gathering and openly sharing resources that scientists and clinicians and researchers of all types need to develop treatments for CANT. Some of these resources could be things such as mouse models or cell lines or natural history study data, to name a few. Now, with therapeutic development defined, how does the process of therapeutic development actually work? In an ideal world, therapeutic development would work like this. You have an idea, A, then do some very streamlined work and eventually arrive at B, which would be a treatment or a cure. This is how a lot of news stories and articles tend to describe the process, but those stories often leave out a lot of critical information because while we wish therapeutic development looked like this, in reality, it looks a lot more like this. Instead of simple and straightforward processes, scientists are often working through a process like this, where there's a lot of testing and retesting, confusing findings, and dead end pathways, but ultimately still moving forward to find answers. Scientists gain so much more information and understanding through this process than they do this process. And it's important to note that the reality, this process, involves a lot of trial and error and necessary failure. But the reason that failure doesn't set us back is because we have so many routes established that we can explore in parallel. After learning more about therapeutic development, next, let's ask, how does giveaway.org specifically address and approach therapeutic development? First, we aim to de-risk canned research and therapeutic development. To de-risk this process, we ask ourselves, what would make potential therapeutic partners hesitate when thinking of partnering with us? And once we can identify a potential barrier, we work diligently to remove that barrier so it's not even on the table. Second, we cast a wide net of opportunities that allow us to pivot quickly. So we aren't just pursuing one therapeutic option, we pursue multiple at a time. This is important because, as I just talked about, some of these projects will fail, and that is okay. We do not fear this failure, and we expect it as part of the process and have multiple initiatives going on at the same time so we can pivot when needed. We also hold a seat at the table of important stakeholder meetings. This can be difficult to do at times, to have a rare disease organization involved in big level conversations in industry or government meetings. But it is so important to have us there to bring the rare disease and community perspective that we can offer. And we are also, of course, relentless in our pursuit for finding treatments and cures for CAN. This is at the root of all we do here at kifone.org and is the attitude that we bring to work each and every day. So, why is our approach unique to therapeutic development? First, we operate with open science principles at the heart of everything we do. This means that the resources we create are open and available to anybody who wants them. So we don't want any limitations holding back progress. We want resources, data, anything that a partner could want access for to help us move to our collective goal of finding treatment and cure. We want that as openly available as possible. Next, we put patients at the center of every conversation, and this is critically important. And we work hard to make this a focal point of discussions in the meetings that we're having. And lastly, we have a dedicated kifone.org science team working with developers. This includes org staff like myself and extends to our over 100-person research network that we've grown over the past few years. So, where are we at now in August of 2022? We currently have multiple active partnerships. And to go over a handful of these partnerships, who are we currently working with? First, a group that you'll hear from throughout the conference are Ovid and Columbia University. They have a great relationship in which they're together working on gene therapy and RNAi focused gene targeting. We also have a relationship with NewSite. NewSite is our partner for our treatment accelerator program that I'll be discussing in a few slides. And with the NewSite, we work on iPSC and neuronal characterization, so learning about certain cell types related to CAN. And we're also developing a drug screening platform with them, so we can have a centralized place where we can test compounds or drugs on canned relevant models. We also work with Biolumix. They are greatly equipped to help us identify new targets and work on screening. We also do single drug repurposing efforts with multiple different academic and biotech collaborators. And how this works is, say, a biotech company or an academic collaborator were to come to us and say, hey, I have this compound or I have this drug that was initially developed for Parkinson's disease or Alzheimer's disease, but I think it could have some utility in a canned model. Well, then we work together to try and test those hypotheses and see if there is relevance in canned. We also work with the Murdoch Children's Research Institute in Australia for their targeted high throughput screen that they're working on, as well as their epilepsy focused screens and projects as it relates to canned. We are also working with teams at the Albert Einstein College of Medicine that have multiple industry partners, such as Atomwise, that allow them to work together to work on artificial intelligence and how it can relate to drug discovery. And we also have a number of new relationships that we are in the early stages of entertaining and developing to make sure that we are entertaining as many opportunities as possible. We want to take a moment to recognize and highlight that some of these relationships and partnerships that were once quite young have blossomed and matured and evolved over the past few years. The MCRI and CMRI team over in Australia was able to secure a massive grant focused on epilepsy treatment for CAND from the Australian government. This was a huge win, and we're so proud of all the work that's being done by the Kifone Research Network members over in Australia. Research at Albert Einstein College of Medicine were winners of the 2022 XSEED Award that focused on identifying drugs targeting the Kifone protein. And additionally, we have concluded our work with Rarebase, their study on ASO administration for axonal degeneration, and we are currently reviewing the data of what they found. And at our conference last year, we presented a brand new initiative called our Treatment Accelerator Program. But as a reminder, what is the Treatment Accelerator Program, or TAP, as we like to call it? The goal of TAP is to create an industrialized and centralized screening platform to test potential therapeutics in preclinical canned models. In other words, we are creating a one stop shop where our collaborators can come to us with potential therapeutic drugs or a specific technique, and we can determine the potential benefit of these compounds in different canned model systems, such as patient derived cell lines. So, how have we done this? How are we doing this? We've partnered with a research team at NewSite, who we've mentioned before, and we will hear from later this afternoon to build this system. And briefly, this project has four steps. First, we create neurons with KIF1A mutations to model or represent this disease in a dish. And we've actually done this. It's important to note that this step was completed by using patient samples to generate these types of cells. Second, we phenotype or collect observable characteristics of healthy neurons versus canned neurons. In other words, because we've completed this step, we can now tell how certain KIF1A mutations impact certain neuronal characteristics. And it's been a great year of progress, and we are pleased to announce that as of this month, we have started screening potential canned drugs in this system to help us achieve our third step of testing promising treatments for canned. And all of these steps lead us to our ultimate goal of identifying therapeutic candidates for clinical trials. And lastly, to catch you all up on where we are at now, we've launched new clinically focused studies that importantly involve you all as part of the therapeutic development process, such as the Koala study and the EEG study. Now, we heard from the Chung Lab about the Koala study earlier today, so let's just briefly touch upon how this study advances our therapeutic development. One of the awesome aspects of the Koala study is that it generates clinical outcome measures and endpoints. What this does is it allows us to determine how effective potential therapeutics will be in our patient population. In other words, the Koala study plays the necessary role of helping us determine what we want to test in a clinical study once a drug or a therapeutic option is ready to be tested in the patient population. The parameters we want to test in patients need to be figured out ahead of time to identify testable endpoints that are effective and meaningful to our patient population. And the Koala study is allowing us to do just that. Most importantly, how are we going to take all of this work that we're doing and bring treatment to our community? We are turning over every stone in terms of potential therapeutic options by working on both short term and long term solutions at the same time. Essentially, we are working on establishing both treatments and cures in parallel. We are exploring everything from compounds at your local drugstores to longer term goals such as gene editing. And to be candid, it will likely take longer to establish a cure than it will be to establish a treatment that can relieve canned symptoms. And so, until we are able to secure that cure, we want to find treatments that can reduce symptoms or slow progression. And while timeline is difficult to determine when it comes to clinical trials, one way we are bringing our community closer to that point is by developing tools and resources that expedite this process. These are things like preclinical tools for development that are useful for our researchers and scientists, as well as patient centered studies like the natural history study and the koala study, both of which depend on community engagement to be successful. Finally, to wrap it up, we want to reiterate that it is our mission to find treatment for every single canned patient. KIF1A is a very tricky puzzle to crack, which is why we are exploring so many options at the same time. But no matter the mutation, no matter the presentation, we are working to find a treatment and cure for all of you. Thank you.